Good morning, everyone. Thank you very much, Carsten, for the invitation. Thank you very much, Ioana, for the introduction. And thank you all for coming at this time in the morning. I'm going to present us a rather recent project on selenium-binding proteins and what that could have to do with nutrigenomics. And I put the SNPs in there, the single nucleotide polymorphisms that were detected in the gene encoding the selenium-binding protein, and I will go from there. But before that, whenever I tell people I'm from Jena, they think I'm from Vienna. Well, they know Vienna, of course, but nobody in the scientific world. Jena is rather well known, but otherwise. So I'm going to just quickly show you a picture of Jena. It's not as beautiful. Well, it's beautiful to me, but probably there's more beautiful areas here in this city, in Olsztyn. Olsztyn is bigger than Jena. Jena is about 100,000 people living there. A large part of the people living there are actually students and professors. So the university has a big role, plays a big role in Jena, and that is also seen in the market square, where there is a statue of the founder of the university. And every PhD student, right after graduation, has to, and that's what my PhD student Dimi does here, try to throw a wreath over the sword. And it takes some time, of course, to reach the sword. And that time is usually used by people standing around and drinking some local beer. So, so much about that. But let's get to the selenium-binding proteins. Selenium, of course, you know, as a micronutrient required for or essential to human nutrition. It is active in humans as part of selenoproteins, of course. And selenoproteins are proteins that carry selenium in the form of the amino acid selenocysteine. That's the 21st amino acid. And the special thing about selenocysteine being incorporated into proteins is that it is incorporated co-translationally. That is, there is a machinery in the body or in the cell that encodes, or that sort of interprets regular stop codon in an mRNA, or reinterprets that stop codon to insert selenocysteine at that position. Once certain requirements are met, such as a selenocysteine insertion sequence in the untranslated region, the 3' untranslated region of that mRNA. But all in all, we have 25 selenocysteine-containing proteins as humans. 25, right? It's not that many. But there's organisms who have even less, such as the nematode Caenorhabditis elegans, has just one selenocysteine-containing protein, but still has the whole machinery of inserting selenocysteine, just because, you know, despite it just being one protein. Anyway, that's what's commonly known as selenoproteins. But there's another form of selenoprotein, and that is could be essentially any kind of protein, because we have this incorporation of selenomethionine instead of methionine, just statistically during translation in proteins, and of course that way every protein containing methionine can become a selenoprotein, now carrying a certain fraction of selenomethionine rather than methionine. But there's another kind of selenium-containing protein, and that's why it's the header, selenium-containing protein. Those are the so-called selenium-binding proteins. They were then identified in 1989 in an experiment using selenite, radioactive selenium-containing selenite. Injected IP, well, this is not IP here, of course, I know, but into mice. And hours after that injection in these mice, they were just looking for what shows up, you know, which proteins show up. And they identified two bands of proteins that were not known then, and that later on were demonstrated not to contain this selenocysteine insertion sequence. At plus UGA, which we know now is essential for selenoproteins to be produced. So all in all, they found it does not contain selenocysteine. It is a non-amino acyl selenium-containing protein, and then they just termed it selenium-binding protein. And the hypothesis was that it just binds to selenite to in some way. In humans, we wouldn't have two bands showing up here, we just have one selenium-binding protein. Now, what is known, or what was known for a while about selenium-binding protein isn't really much. You know, it wasn't even known how selenium binds, or it still isn't really known how it binds to the to a selenium-binding protein. Is it, you know, through a selenosulfide, which was postulated for the human protein? Is it this kind of sulfur-selenium-sulfur type of binding of selenium to this protein, which was hypothesized for plant selenium-binding protein to be the case? It's really not known. What is known for the human selenium-binding protein is that it is a 52 kD protein expressed, you know, pretty much ubiquitously, but at highest levels in the lung, in the colon, and the liver. And it is usually a cytosolic protein, but we now know that's not always true. About the regulation of its expression, also not much is known. There's some transcription factors that were identified, including hypoxia-induced factor 1 alpha, and also the significance and the function. Not much was known. It was hypothesized to be a tumor suppressor because it was shown to be low levels of selenium-binding protein, to be low in human cancers, pretty much all over the place. And usually there's also a correlation with the prognosis of that cancer. Once selenium-binding protein levels are low, there's a bad prognosis in terms of outcome of that cancer treatment, etc. And well, selenium-binding protein was also hypothesized to be a marker of differentiation status of cells. But regarding the actual activity, well, that's about as much as we knew at that time. At that time means 2017 maybe. Regarding this differentiation marker, I'm just going to give you a brief example of what this could mean in terms of selenium-binding protein being a differentiation marker. We analyzed the differentiation. It's just a cell culture type of experiment of 323 L1 pre-adipocytes into adipocytes in cell culture. And along the differentiation period of 2 weeks, we see, of course, the upregulation of certain transcriptional regulators that are known to be regulators of lipogenesis, of a protein that is known to be attached to lipid-containing vesicles. And then, of course, we also saw this upregulation of selenium-binding protein, not only at protein level, but also mRNA level, and it pretty much correlated with lipid accumulation in these cells. We then went on to look at what regulates this upregulation, found some further factors that control this, including, for example, mTOR, this mammalian targeted rapamycin kinase, which is a known sensor of energy and building block sufficiency status, and this inflammatory marker, inflammatory cytokine TNF-alpha, those were identified as regulators of this process. Here you can see these lipid droplets in these differentiated cells, and you see in yellow here the cytoplasmic localization of selenium-binding protein in these differentiated adipocytes. We also tested this in a mouse experiment where mice were fed a fructose high diet and were tested for non-alcohol induced fatty liver disease, NAFLD, and those mice did have NAFLD, but what we saw is that there is not really the same correlation in these fatty livers now, right? So we cannot say that there is always this correlation between lipogenesis and selenium-binding protein expression. So that's about as much as we knew at that point. At that point, again, it was 2017, 2018. Now, before I get to 2018, maybe let's talk a little bit about lunch. You know, if you go for lunch, you could choose, for example, to eat asparagus, and everybody knows what the problem with asparagus could be. And you know, sometimes just hours after consumption of asparagus, is that some people apparently wish they had a mask protecting them from the odor of what's coming out of people having eaten asparagus. And there was actually a genome-wide association study investigating this phenomenon, published in 2016, with rather strange outcomes. Strange, I have to say, because the numbers that came out of it really seem strange to me. But let's get to it. What they investigated was the ability to smell asparagus metabolites in urine of people. And this was essentially just a questionnaire that was sent out to close to 7,000 participants that responded. It was sent out to more than these 7,000 of already existing cohorts, right? So they had the genetic information, and they had then the answers, well, to the question, do you smell asparagus? And does it smell bad to you, or do you not smell it at all? And the non-ability to smell this asparagus-derived odor is called anosmia. Okay, so they differentiated between asparagus smellers and anosmic people. What they found is that about 60% of the people returning their questionnaire were anosmic. See, that's what's strange to me. I know about just one family in my whole surroundings, everybody I asked, just one family who don't smell asparagus. Everybody else does smell it. Everybody else I have asked. Of course.
How many people smell asparagus?
Oh yeah, good idea. Maybe the other way around. Who does not smell asparagus? Oh, okay. 2 more I know now. That's good. But it's definitely not 60%, right? So that's what's puzzling me. And so I thought maybe it's about a degree. Sometimes people just smell it but don't feel like putting up a gas mask, right? It's a degree of how bad you feel about that smell. But then they traced it down to something. As they knew their genetic or had their genetic information, they pretty much traced it down to single nucleotide polymorphisms in the genetic information for olfactory receptors. So it is not the capability of metabolizing asparagus, but rather of sensing the asparagus smells that comes out of here. But why am I into asparagus? The question is, what is this odor? What is that stuff that you smell? And it is largely these metabolites here. It's all sulfur-derived metabolites that you can smell, including, for example, methane thiol. And this methane thiol, simple molecule, actually smells like cabbage or rotten fermented cabbage, or kimchi, maybe the Korean kimchi. That's the version of cabbage they use, but cabbage is very frequently consumed all over the place. And certain cheeses, of course, they also exude that smell of methane thiol. And that's what I want you to keep in mind, because now I'm getting back to selenium-binding protein. There were, and that's now 2018, in a study published in Nature Genetics by a Dutch group, they report on results they had in the Dutch hospital of 5 patients from 3 families. Some parents were consanguineous in these families, and the common symptom they had was cabbage-like malodor, and it was actually pungent, described as pungent breath malodor. And this being cabbage-like malodor, they tested for sulfur-derived or sulfur-containing compounds, and they identified methane thiol and breakdown products, methylated products of methane thiol in their odor. And the question is, why do these patients have this content in their odor rather than, everybody else? And they traced the whole thing down, and that's the intriguing story, traced it down to mutation in the selenium-binding protein 1 gene. And then they followed up on what is going on here. What does this selenium-binding protein 1 do with these sulfur-containing compounds? And they found it to be a methane thiol oxidase, breaking down methane thiol before it can do anything to whatever parts of the body. The prevalence of this was estimated, it's of course just an estimation because it was only few people, 1 in 90,000. It was then termed a rare variant of extraoral halitosis. Halitosis being bad smell, breath, odor. And extraoral halitosis means the source is not in the mouth region, but elsewhere in the body. So selenium-binding protein now as a methane thiol oxidase, and this is the reaction that it catalyzes. Methane thiol is oxidized to form, well, formaldehyde, hydrogen sulfide, and hydrogen peroxide. And I'm marking these two, hydrogen sulfide and hydrogen peroxide, well, because for one, they are signaling molecules in the body, but second, we can use those to detect methane thiol activity. And that's what we went out to, but here's, before I do that, a summary of these findings, right? You have methane thiol normally, which is broken down by selenium-binding protein 1, which is expressed in the colon area, well, H2S, hydrogen peroxide, etc. Whereas if you don't have selenium-binding protein 1, active selenium-binding protein 1 that is, you exhale methane thiol plus methylation products such as DMS, DMSO, and DMSO2. Now, as I said, we use this H2S and hydrogen peroxide now to establish an assay, because we're a simple biochemistry lab, right? We don't have a gas chromatography right around the corner that we can use anytime for a number of samples, so we thought about a way of detecting it simply in the lab, and each day we just want to do it. I mean, the consequence is that our lab from time to time smells like cabbage, which is okay sometimes, but you don't always like cabbage in the morning. Anyways, so we thought of the following assay. And the assay, or the thought was that because we cannot just purchase methane thiol, I mean, of course you can purchase it, but it's poisonous and you have to take extra care, and it's a gas, and it comes in gas, containing containers, and etc. And since we're just a simple biochemistry lab, we had to think of a different version of dealing with methane thiol, of creating methane thiol. And the thought was, let's create it in situ, biochemically. And that's what we did. The thought was, we just use methionine, well, regular amino acid, and imitate the situation in the colon. Because in the colon we have bacteria that have methionine degrading enzymes, and the product of this methionine degradation through methionine gamma lyase activity is methane thiol. And then let's detect this methane thiol oxidase activity by assaying hydrogen sulfide and hydrogen peroxide, which are rather simple to detect. And this is the setup that we came up with. It's a really simple setup. Essentially a microtiter plate. We fill some wells with the methionine gamma lyase plus methionine, of course. It generates a methane thiol, which gets into the overhead space here underneath the lid of this microtiter plate, and it gets into just diffuses into the next area, which contains the methane thiol oxidase, or any cell lysate that we think might contain a methane thiol oxidase, etc. And then we just detect hydrogen sulfide and enzymatically detect hydrogen peroxide generation. And this is the generation of this setup. We can, of course, first demonstrate by gas chromatography that methane thiol is really generated, and we can see also that there is hydrogen peroxide generation in this setup. So, bottom line, it works. And this is what was demonstrated now using selenium-binding protein that we generated. We expressed it bacterially, isolated it, and then we had isolated selenium-binding protein. We also used these single nucleotide polymorphism-derived variations of selenium-binding protein that were described in this original paper from this Dutch group, which we knew from that paper should be inactive, so that's our control. And then, of course, we saw what was expected. We see the hydrogen sulfide generation in our selenium-binding protein 1 containing samples, and we see the hydrogen peroxide generation here, and it's well in the linear range, so we can trust these data. And in the mutants, described in those patients from that Dutch hospital, in the mutants, we didn't see any hydrogen sulfide or hydrogen peroxide generation anymore. So much for the assay, and that's what we were intending on using now, to get to what selenium-binding protein actually does and what's the significance of it might be. So let's get back to the question of differentiation in selenium-binding protein 1. But this time, maybe a different model system, because we now know that the colon might play a role in the microbiome from these hospital patients. So we used CACO2 cells, colon cancer cell model that has the ability of differentiating to colonocyte-like cells. Once you keep them in confluence, under a confluence state for at least 2 weeks, right? And then they spontaneously differentiate to this colonocyte type-like cells. And then we looked at what's going on with selenium-binding protein. Selenium-binding protein is upregulated in these differentiated CACO2 cells relative to the non-differentiated or just confluent cells under these conditions. And that comes with an increased activity, of course, as we can see here, H2S activity and hydrogen peroxide generation both go up. So we can use this assay for cell culture experiments, and we later showed that we can use it also for samples from animal tissue, for example. But yeah, what does this mean in terms of differentiation? We see an upregulation of selenium-binding protein once we have differentiated colonocytes. I mean, what would they represent in vivo? And then we got back to the literature and found these examples here of something that was already known for selenium-binding protein expression. And as you can see here in the colon, you see the highest expression of selenium-binding protein at the tip or the apical side of the crypts. Just to put this into a cartoon, this would mean the following. You have the crypts here with the undifferentiated colonocytes, and going up into the top of the crypts, you see an increase in differentiation of these cells, less proliferation, of course, but we also now see an increased expression of selenium-binding protein 1. That's the blue dots over here. And what happens with selenium-binding protein 1 here is that it encounters the degradation products of the microbiome, including methane thiol. And this methane thiol now is degraded to form formaldehyde, hydrogen peroxide, and hydrogen sulfide. Apparently, the cell can deal with these rather easily, different from methane thiol. Here, maybe as an aside, remember I told you there was this hypothesis that selenium-binding protein could be a tumor suppressor, and the evidence for that is well, circumstantial. It has low levels in human cancers, and it is upregulated in differentiated and downregulated in maybe dedifferentiated cells. And there is this association, I mentioned this before, with a poor clinical outcome if you have low levels of selenium-binding protein 1, which would mean that you could have an exhalation of methane thiol and methane thiol degradation products under these circumstances. And this is actually why it was hypothesized that this could be a reason why there is, maybe we should ask around, cancer-smelling dogs. And the PhD student doing these studies into selenium-binding protein actually contacted a person working in this direction, and it did not seem absurd to draw these conclusions to that person. Okay, anyways, we now know that there could be something with regard to methane thiol activity, with regard to cancer relation, etc. But how can we get into further detail? And maybe within the confines of a PhD thesis, right, which you know is about 3 years, maybe a little longer sometimes, and that makes mouse work difficult, right? Creating a mouse, a knockout mouse or something. So we thought of a different model that we're using in the lab, and that is Caenorhabditis elegans. It's a worm model here, and we use it because of various reasons. For one, it's really cheap and simple to handle. very much cheaper than mice. You don't have to, get ethics permission to work with C. elegans. It's so you can work with it faster. And if you have any idea, you can just change your setup. So that's one reason. It's transparent, so you can look at changes under the microscope. And short-lived, that is, you can do lifespan studies within the confines of a PhD thesis, right? It lives just 30 days, so you can do several studies that way as a PhD student. And, well, the most important point to us is, of course, there's a huge number of orthologs in the genome of C. elegans to mammalian genes, right? The classic example would be the insulin signaling pathway. The whole pathway is the same, pretty much the same in C. elegans as it is in mammalian cells. So that's an example of these orthologs existing between C. elegans and mammalian situations. So we looked at C. elegans. Does it have a selenium-binding protein? And nothing was known at that time about selenium-binding proteins in C. elegans. So we looked at the databases and came up with two potential selenium-binding proteins with very high identities, not homologies, identities of more than 50 and more than 30%, with similar molecular masses as the mammalian one. And then thought, well, what about the expression? What about the effects of these proteins on lifespan, stress resistance? What about MTO, methane thiol oxidase activity in these worms? So what we did is we genetically modified. I mean, that's another reason you can use C. elegans easily, because it's easy to modify or manipulate genetically, and generated worms expressing a promoter and open reading frame containing a GFP-coupled version of this first selenium-binding protein that I now call, for sake of simplicity, just Y37. And we looked at where it is expressed, and we found that it is expressed in organs or regions of the body that deal with energy metabolism and fat storage, and also development, and that are in the vicinity of the environment, which is important because the environment of C. elegans contains bacteria, which, as we know, produce methane thiol. We looked at the expression levels of this Y37 over time in these worms, and close to after adulthood, so close to their dying after 30 days, you have lower levels of selenium-binding protein 1. The other one, this R11 protein, turned out, and we generated worms in the same way, overexpressing a GFP-coupled version. That is really weird, because this protein was expressed only in 2 neuron pairs. It's a strictly neuronal potential selenium-binding protein ortholog. Strictly neuronal. And speaking of neurons, we know that C. elegans has about 300 neurons, and overall it has about 1,000 cells. So 2 neuron pairs is 4 cells in total that express this selenium bone, perhaps a potential selenium-binding protein ortholog. It was found in neurons that are known to be, for example, important for temperature sensing of C. elegans. Okay, so what about the MTO activity, the methane thiol oxidase activity of these proteins? And we used C. elegans lysates, and we used the isolated proteins. Again, we overexpressed the Y37 and the R11 proteins and did our assay in vitro, and we did it with worm lysates. And we found that the Y37 really has a very high methane thiol oxidase activity. And that's why we gave it the name SEMO1, which is, it's a selenium-binding protein ortholog with methane thiol oxidase activity. So SEMO1 is this selenium-binding protein ortholog in C. elegans. And we looked at the other one, the R11, and it does not have any methane thiol oxidase activity. That's why we called it SEMI1, selenium-binding protein ortholog with MTO activity. MTO inactive. And if we look at worm lysates with knockout worms that we generated or had generated, And we see that if we knock out the R11, of course, there's no loss of overall MTO activity, but if we knock out the Y37, the SEMO, we have a complete loss of methane thiol oxidase activity in worms. So now we know that there's selenium-binding protein orthologs in C. elegans, and we can go from there, right? We can use it as a model to investigate significance of selenium-binding protein. And what we did is we looked at lifespan. It's an easy thing to do with C. elegans, right? You can check if a protein is important for aging or for lifespan and stress resistance. We found the following interesting aspect. We found that in worms without SEMO1, that is knockout worms, we have an extended lifespan and an extended, not shown here, stress resistance. Oh, actually it is shown here down there. An enhanced stress resistance against oxidative stress derived from paraquat exposure. It says 300 millimolar paraquat. It's a, that's a really high concentration, but you have to think about the setup. It's just 300 millimolar in an agar plate, and the worm just takes up fractions of that, so it's much lower in the worm, of course. And so we saw this increase in lifespan and in stress resistance, and the funny thing is, we saw the same thing in the SEMI-1 knockout worm, the one without the methane thiol oxidase activity. Apparently this, or to us, this implies that the methane thiol oxidase activity might not be essential to this lifespan or stress resistance effect. But we will see about that. The question, however, is if this worm contains a protein, or let me just read it, can afford to produce proteins that shorten lifespan and lower stress resistance, these proteins must be important to the organism, right? The question is then, what is the biological role of these proteins in the worm? I know we're digressing from the human, from the mammalian system, but now we got interested. I mean, what is this good for in the worm, in the model that we're using? And again, the thought is we have an extended lifespan when the protein is missing, which means the protein itself must be shortening lifespan. And why does the worm produce this protein? And what is it good for? And what we came up is the following. Not only is it an MTO, a methane thiol oxidase containing protein, which is important because of the worms being in an environment producing methane thiol, but also we came up with the result that it apparently serves as a selenite buffer, conferring something like a selective stress resistance. Remember, the resistance against oxidative stress was increased when the protein is missing, but the resistance against selenite was decreased when the protein is missing. So quite the contrary. And this decrease, or the selenite sensitivity increase in the knockout or in the knockdown worms, led us to conclude that apparently it works as a selenite buffer, which makes sense, right? After all, it's called selenium-binding protein. And selenium, of course, is contained in the environment as well, because the environment is just regular soil, and soil contains inorganic selenium, for example, selenite, selenate, etc., to certain degrees. And so, all in all, this would probably help the worm to survive long enough to reproduce. And after reproduction, it just doesn't matter if the worm lives shorter or longer or whatever. And this is what I meant by the environment. Usually they are found in the soil. These worms, 1 millimeter in length, these worms, you find them in dung, etc. And so this is what the worm would be exposed to, for example, methane thiol from root damage, found down here, or a biodegradation of methionine, for example, or sometimes nematicides are used that contain sulfur, such as DMDS, dimethyl disulfide, was demonstrated to have nematicidal activity, and that would also cause the worms to be exposed to methane thiol. That would be a protective effect then, of course. This protein expressed close to in organs close to the environment, contains this active protein. Now what about the other one, the neuronal one, without the MTO activity? Remember I told you it's expressed in neurons that deal with temperature sensing? And we tried to test that. So a PhD student, Wei Gong, came up with an idea to test the temperature sensitivity. It's a really simple setup. We have an aluminum plate here, we have a hot area on one end and a cold area on the other end, and we just test using a thermometer, laser thermometer, test the temperature gradient across this aluminum plate, and then put worms in the center, and then just look to what temperature do they travel, essentially. And we know that their optimum temperature for breeding and for reproduction, etc., is around 20 degrees. And so all in all, they usually travel or grow. I don't know if you can really see this, but they went in this experiment, they went to the right of the plate, which is the goal, because that's the optimum temperature in the wild type worms. And we have a control mutant, and these control mutants, devoid of temperature sensing, there is no such thing. And so what did we see in worms devoid of our R11 protein? Well, we saw a similar thing. We saw that there is less thermo sensing, less thermotaxis in worms devoid of semi-1. The conclusion would be that R11 is, or semi-1, is required for positive thermotaxis. And we also see the same thing in negative thermotaxis. Negative and positive just refers to the way of the, movement on this in this gradient. Wherever you put the worm, you always want to get to the 20 degrees optimum temperature. So semi-1, which is now the official name, semi-1 is required for proper function of these neurons. Okay, so much for C. elegans. Did we answer any questions regarding the biological activity or the significance of this protein? Well, maybe we know something about it being a selenium buffer. We know, of course, of its methane thiol oxidase activity, which had been known before. We knew or now know that it does act as pro-aging and factor and does modulate stress resistance. But what does that tell us about the mammalian protein, which is the ultimately interesting protein that for us? So we went back to the selenium-binding protein one, the mammalian one, and we answered the following questions. First one being, is it specific for methane thiol? How about other thiols? And we found that it does have the highest activity, or is methane thiol is the best substrate, highest activity with methane thiol and ethane thiol. But it is also active with other thiol-containing components that we tested. So we can now say that selenium-binding protein 1 is not just a methane thiol oxidase, it's an alkyl mercaptan oxidase, because there's several substrates that are being oxidized when under the catalysis using MTO. And these are the examples that were tested: methane thiol, ethane thiol, all found in nature. Methane thiol, I already told you about, cabbage. Then there's ethane thiol, propane thiol, all found in plants and skunks and whatever. All of these are stinky metabolites, essentially, thiol-containing compounds. And all of these are substrates to this protein, to selenium-binding protein 1. How about the other questions? Is selenium required for this methane thiol oxidase activity? I'm going to cut things short here now because of the time. No, it's not required. It's independent of selenium. So selenium, does it bind to the protein at all? And the answer is, well, only at very high concentrations. There is some binding of selenium, used in the form of selenite here, to that protein. 0.3 selenium per protein molecule was identified by us at really high concentrations of selenium. I mean, if you look back to the original experiment for the identification of this protein in mice, that's what they did. They injected IP, high concentrations of selenite, radioactive selenite, into mice, and then just looked at what's going on, what's showing up as radioactive selenium-containing proteins. And, well, apparently they were lucky, because under normal circumstances, you wouldn't have seen such a signal, because you have this very weak, apparently, selenium binding. And selenium binding was always hypothesized to be via interaction with cysteine residues in these proteins. Well, it's got 10 cysteine residues, and we created various mutants of these proteins, and none of these mutants had any major loss of activity. So apparently the term selenium binding protein is not really appropriate. Maybe it really should be called a methanethiol or alkyl mercaptan oxidase after all. But, and that's the last point of this presentation, if it doesn't bind selenium, what does it bind? And where does the MTO activity come from? What kinds of oxidases are known in biochemistry? And there's two major types of oxidases. There's FAD derived from riboflavin, FAD containing oxidases, and there's copper-dependent oxidases. And that's what we investigated. Again, using our assay that we set up, and we tested for transition metal ions and their effect on the activity. And this was actually a weird coincidence, because two PhD students, isolated the protein, and in the hands of one student it was active, in the hands of the other student it was inactive. And what we found, we looked at that, it's a happy coincidence, one could say, and the only difference between the buffers used was EDTA. One used EDTA, the other didn't. And that's how we came up with this idea that the EDTA could be the reason. And that's what you see here, actually. You see this protein activity, hydrogen sulfide and hydrogen peroxide generation in the presence of EDTA is down. And then the question is, how can we recover activity, right? We used EDTA to have it go down, and then added transition metal ions, and the only transition metal ion recovering and more than recovering activity was copper. And then we did the same in C. elegans. Got to be good for something that we have this knockout worm, and we found the same thing. If we grow the worm in the presence of higher copper concentrations, or grow the worm in the presence of a copper chelator, such as bathocuprine sulfonate, BCS, and then lyse the worms, what activity do we see? And we see an increased activity in the presence of copper and a decreased, very, very low activity, methanethiol oxidase activity in these chelator-generated worms. And of course, there's no such activity at all in the knockout worms. So the summary would be, it is a copper-dependent enzyme. And we then did further mutation studies in order to identify how the copper is bound to the active site. And to that end, there's no structure available. There's only a structure somewhere for a bacterial enzyme. So we went back to the AlphaFold, or the structures available through AlphaFold and Ki-generated potential structures. And we looked at what's given over there, and we found at the potential active site some histidines and an aspartate and a glutamate, all amino acid residues known from copper protein or copper biochemistry to potentially chelate or bind copper ions, right? And we mutated any of these out, and whenever we mutated any one of these out, we saw a complete loss of activity. So that we hypothesize that actually, not only do we see a loss of activity, we also see not a complete, but to some extent a loss of copper binding of these proteins. So that bottom line is that both the human and the model organism, the C. elegans protein, are copper-dependent oxidases. And we know that two histidines, a glutamate and an aspartate, are the chelating amino acids. Again, the structure is unknown yet, still unknown. We also tried EPR studies to see what kind of copper protein this might be, but to no avail at this point. We're trying to stick to that, but that's the major conclusion. So, to sum this up, I've told you about selenium-binding protein 1, identified as a methane thiol oxidase in a Dutch hospital with these patients with the breath malodor. And that was the initial point for us to try to establish a simple methane thiol oxidase activity, to be able to test this in a regular biochemistry lab without any gas chromatography and NMR, etc., around the corner. And then I also told you about selenium-binding protein and its role in differentiation, especially in the colon. And we saw that there is the highest levels of selenium-binding protein in the most differentiated regions of the colonocytes. In C. elegans, which we use as a model to get deeper into the function of this protein, we saw that, first of all, C. elegans does have such a protein, which had not been known at that point. So we can use it as a model. We saw that there is something like lifespan and stress resistance regulation by these orthologs shown. Well, we now know that there's 2 orthologs and that they might be important for the biological situation of C. elegans, for reproduction, etc. In what way still needs to be defined. But getting back to the human selenium-binding protein, we know that it is an alkyl mercaptan oxidase, implying that it has other substrates than just methane thiol, and that it is a copper-dependent oxidase. So all in all, the talk was called From SNP to Function. All in all, it all started out by identifying single nucleotide polymorphisms in genes, in the gene encoding this selenium-binding protein, ending up in a completely interesting and different activity of this protein, different from what had been anticipated, in it being a methane thiol oxidase. And the significance of this methane thiol oxidase needs to be defined. I mean, the question is, in humans, is this methane thiol oxidase activity really required for differentiation processes? We don't know. Same in C. elegans. Is this methane thiol oxidase activity required for lifespan effects? We think it's unlikely because there's this other worm with the semi that doesn't have any lifespan or doesn't have any MTO activity changes. But anyway, so does this mean that there's if you have bad breath, you have a longer life? You know, that would be the conclusion from the C. elegans studies. And of course, no, it's just a worm after all. Right? The worm, for example, is not just your father, it's also your mother. It's they're hermaphrodites, right? Only 1.1% in the population are really males. But still, you can think of what could this mean, and if you translate the experiments, we know that there is a lifespan extension if you have a lower level of SEMO1. Worms don't get cancer, you know, but you know, the lifespan of a cell, what could this mean in terms of the human situation? It could mean that by down-regulating selenium-binding protein 1 levels, you could somehow, and you know, induce de-differentiation of cells, maybe increasing their proliferative activity, their lifespan in a sense, imitating the cancer, that's a situation that you have with cancer. But that's, you know, that's just a hypothesis. It's not really known if you can do this kind of translation from worm to humans. And it's just more than a hypothesis, it's a speculation, to be honest. Anyway, I'd like to thank the people involved in this study. First of all, it was in Thilo Philipp, a PhD student just finishing up. He's moving to Switzerland now for his postdoc, but he did all the studies on the isolated selenium-binding protein, on the assay, on the copper binding, etc. And then there's Wei Gong, well, looking out here in the end, but closer here, who did all the worm studies with the neuronal selenium-binding protein. And, well, what you see here is, I mean, that's a good thing about Jena. There's vineyards close by. And that's what we do, you know, a retreat every now and then. We test the local wines. But on the way there, my car, I got lost. We ended up, funnily, in Wormstedt. So that's the worm people from the lab, and they were in my car as well. So when on the way to the vineyard, we ended up, that's quite nice over there. So Thilo Philipp, Wei Gong I've introduced, and Holger Steinbrenner, of course, who's a colleague in the lab as well, and who did all the studies on the differentiation in adipocytes, and also works with the PhD students and so forth. And there's other students mentioned over here, Verena Ose, for example, it's her. She does the studies on copper oxidase in worms. And there's collaborations with, for example, Martin Strako in Edmonton, who identified the neurons for us. He's a specialist in C. elegans anatomy. And Antonio Miranda Vizuete from Sevilla in Spain, he did the injections of the plasmids created for us into the worms for us to generate the GFP-containing mutants. Okay, so I thank you very much for your for listening.